I'm ready to welcome Peter Holman. Peter, where are you?
Hello,
There
Devendra.
he is. Good morning, Peter.
Morning.
I have the profoundest privilege of calling upon Peter Holman, who is the Vice President of Quality at Arsenal Bio, to be the moderator of our next panel, which deals with all those things that we talked about this morning. A lot of it this morning was focused on Apheresis and catering to the voice of the customer. And in my humble opinion, here is an executive who was part of the launch of COMMERCIALIZATION, the first cell and gene therapy product from Novartis. But more importantly, I know of no one who understands the Apheresis landscape globally more intimately and empathetically as Peter, because in his former role as the senior director in Novartis, he was responsible for the global cell and tissue operation, the job being one of integrating Apheresis across, 10, 12 countries in the world. May I call upon Peter to lead the charge with our panel. And on the panel, we have Don Henke. Don, please join us. We have Olive, please join us. We have Greg Bodner from Be the Match, please join us. And we have Gary Hutchinson from Modality Solutions, please join us. There you are, Peter. Take it away, please.
Great. Thank you so much, Devendra. And I'm going to share a screen. I prepared a little presentation slide deck to help walk us through the panel and the questions. Are you able to, if you can confirm if you could see the screen that we have up here?
Sir. Yes, sir, we can.
Excellent. Make sure I can advance here. How do I do that? There we go. So thanks. I think I'll introduce myself, but maybe the first thing is to maybe introduce our panel. So maybe some self-introductions would be good. So Dawn, if you could go first.
Hi, I'm Dawn Henke. Thank you so much for having me here. I'm the Senior Technical Program Manager for the Standards Coordinating Body. So we're a nonprofit that works to help facilitate the development of standards for cell therapy, gene therapy, and tissue engineering. I've been there for about 3.5 years now, a little over 3.5 years, and I help run all of our scientific and technical standards efforts and bringing together and helping facilitate those conversations and helping accelerate the development of standards.
Thank you, Dawn. Welcome. Gary.
Hi, everyone. My name is Gary Hutchinson, President of Modality Solutions. We're an engineering firm focused on doing the design and validation work on the cold chain. Started 10 years ago in the biologics, focused primarily on monoclonal antibodies, and in the last couple of years have started really to get a chance to help out on some CAR T cells as well. Excited to be here.
Thanks, Gary. Olive?
Hi, I'm Olive Sturtevant. I'm the Administrative Director of the Cell Manipulation Core Facility at Dana-Farber Cancer Institute. Our facility serves as a CMO, not just for our adult and pediatric bone marrow transplant programs or the other cancer patients that we treat as part of the Dana-Farber, Brigham and Women's, and Boston Children's collaboration, but we also manufacture products for a variety of 23 plus other clinical sites around the country and manufacture for 16 plus other biopharma companies around the world. And these actually range, these investigational products run the gamut of phase 1 through 3 and could be immune effector cells or gene therapy cells, such as for SCID or beta thal or sickle cell. We also manufacture regenerative medicine cells for ALS, MS, diabetes, etc. given all this, it gives us a unique perspective of dealing with many different clinical sites around the country, but also dealing with a lot of different biotech companies. While we manufacture a large variety of cell therapy products, we also ship out a fair number of immune effector cells out to a variety of biotech companies. So between the clinical sites we interact with and the biotech companies we interact with, we see a large variation of issues that come up, and that those challenges sometimes hopefully we can be part of a good solution to help everybody in this field.
Great, thanks, Olive. And Greg.
Yeah, hi, I'm Greg Bodner, and I lead the client engagement department at Be the Match Biotherapies. And similar to Olive, we get the pleasure of working with a lot of different manufacturers, as well as apheresis centers in this field. Be the Match Biotherapies is a department within Be the Match, which has been around for 30 plus years in stem cell transplants. We've done over 100,000 of them. And so, 7 years ago, the leadership decided to extend these capabilities into the burgeoning, the beginnings of the cell and gene therapy company. And we've been working to do that. And we realized early working with Peter and others, these standards and best practices were so very, very important. So, we think this is a very important area to do and one of the greatest challenges that we have. So I'm very happy to be here. Thank you to BSMA and Devendra for doing this. And thank you to Eileen and Matt for the kind words.
Thanks, Greg. And I'm Peter Holman. I'll be the moderator for this panel. And thank you, Devendra, for the introduction. A little more background on my part. I am fairly new in my role as the Vice President of Quality at Arsenal Bio in the San Francisco Bay Area. I have just transitioned from Novartis, where I was global head of essentially apheresis integration, cell operations, tissue operations, where we partnered with University of Pennsylvania to bring tisagenlecleucel into Novartis, and went through our clinical trials in the early 2010s and launched commercial product in mid 2010s. And when I left 3 weeks ago, at this point, we had 300 hospitals globally with field teams supporting those hospitals. So we had an enormous amount of growth, and I've seen an enormous amount of growth both within Kymriah, tisagenlecleucel, and within the field. So I've interacted with everybody on this panel for quite a while in different functions, and it's great to have the group. So the way we're going to do this, we've got some sort of prepackaged questions that we'll do some roundtable. I feel like I'm a game show host here. I should have a long microphone or something. So we'll have some prepackaged questions, and then we have reserved some time towards the end of the hour to have some question and answer from the audience. And I do have the chat open here as well if there's any questions as we go through. So without further ado, our first question is to the panel is, what do you see as the biggest challenges to the field when it comes to the supply of the autologous cellular material as starting material for manufacturing? And I might actually pick on Olive first, as she's coming from both as a manufacturer and from a hospital perspective. I think it might be nice. And I actually have a slide that Olive had pre-prepared with some of your points. So I'll go to that, Olive, and let you take the first stab at answering.
Okay, thank you. And I think my answer may change from day to day as different issues come up. Because normally I would say, most people that know me, I'm going to actually talk about chain of custody and chain of identity as one of the biggest concerns, especially with dealing with the autologous products. But one of the things that actually has kind of come to the forefront, and I see a bigger issue with, is actually getting the right cells collected from the apheresis material. And some of that might be due to the fact that maybe just having a disconnect or not having the right information given to the apheresis centers, where, a manufacturer may say, I want X blood volumes, but really not looking at, how are they actually collecting, what layer are they actually pulling off of the apheresis material. I do think being more specific and saying, I want X number of MNCs or X number of CD3 cells, and being able to quantify that on the collected material going out to a lot of the biopharma companies may actually help with more success of actually manufacturing the product that you intend to manufacture. But I will jump back to COI and COC. Being an academic center, being a FACT accredited center, we do follow the ISBT 128 labeling practices, as do all the accredited centers. So I've been very vocal about accepting this as a standard of practice for both the outgoing material and the material coming back, especially having very specific information on there. I feel it's very important if you are dealing in the autologous realm that you have to maintain the identifiers that the clinical centers are going to use. So the product can go out, get manufactured, come back and go to the patient without needing to be relabeled, re-identified. And many of you on the call who actually from the biopharma company that have actually dealt with me, we generally spend a lot of time in contract negotiations and quality agreements to ensure that we get the information we need back on our products. I think the other areas that certainly create some havoc sometimes are, dealing with a variety of web portals and the variety of audits and how different people, different companies will focus on different things. I think the least issue right now is really the shipping and even some of the documentation with shipping. I think that has become very standardized with most companies and seems to be going quite smoothly. And I'll let other members jump in now.
I'll go ahead. I think one of the biggest challenges we see right now is, and we just had a little example of it, is communication about what are the efforts that are going on around best practices and standards. I know, even within my own organization, people don't know about the cryopreservation effort that's going on by PDA. So I think there's a lot of great stuff going on, just people don't know about it. And I think we need to communicate that more and get that out to everybody so they're aware of what's going on and what's coming. Then I think it's adoption. I think that's going to be a very big challenge. There is a tough business model to give to somebody, especially a senior executive, to say, hey, I'm going to add risk into this equation with change. I'm going to add cost in this equation. You're going to have to refile with the FDA and change some things. And oh, by the way, it doesn't really necessarily improve the product. In fact, it could maybe reduce patient outcomes. And it's a very hard business model to get across. I think everybody agrees that this path we're on is unsustainable, but really selling that picture and then really walking us through that pathway, how this is going to work, and make sure every company understands, yes, everyone's adopting this, everyone agrees to do this, understanding that these different efforts, which will not come at the same time, they will come in stages, are going to be done and going to be adopted. And some you might be closer to the new standard or best practice, and others might be a stretch for you to get there. But without that, I'm worried about how we get to this point where we all agree we need to be, where we are working together. Because at the end of the day, the competition is there. It's at least for the most part limited by indication, so it's not as big an area of competition. But I think it is a really challenging problem because there's great work that SCB, BSMA, PDA, all these groups are doing. But how do we get these actually adopted?
Yeah, I think I completely agree with both Olive and Greg's points. I think those are all major things that I hear all the time as huge barriers and issues. I think that also you see that some of the communication between the centers, like Greg and Olive have both been saying, could be improved in that oftentimes there's little differences in the cell collection procedures that really could be standardized across the board, because it becomes a huge burden on the centers when they have multiple different manufacturers changing some of their procedures or asking for specific things in the procedures that are really minor changes, but then that means requiring different training, different, remembering all these different procedures, all the different documentation, and some of standardizing some of those simple things, like the things that really don't need to be that different, would really help with that communication barrier, because that communication could become more harmonized and standardized, between both parties and across the whole field. But also, it can help prevent errors and mistakes from happening, because you don't have to have, those multiple trainings and those switching back and forth between SOPs and those variations, and also maybe some sort of standardized way of really highlighting and showing those differences that do need to be there, so that they stand out a little more and are a little easier to understand coming from the manufacturers to the apheresis centers as well. I think that's also a huge barrier and issue that I hear frequently.
Thanks, Dawn. I thought I'd also reflect, thinking back to also Tim's keynote at the beginning, and he talked a bit about challenges in manufacturing, right? From apheresis and questions about maybe different media is better for growing cells from different patients. And I think that's where it is important that we talk and we try to resolve some of these questions on variability that we're putting onto all of the hospitals with so many different manufacturers and more manufacturers coming and wanting lots of different things. But also having seen that there's a lot of variability, even as it is, between all of the different hospitals who are doing things, apheresis centers who are doing things slightly different, which even without different manufacturers putting requirements on, end up resulting in a huge amount of variability of the apheresis material coming in. So not just patient intrinsic differences, but actually process related differences. And I think some of these harmonization and standardization concepts actually can hit both of these and actually simplify things for the manufacturers and potentially for the hospitals. And I think one of those hurdles that you pointed out, Dawn, however, is that sometimes some of these things are filed, right? They may be in INDs, depending on the country and the region. The apheresis centers may have filed, right? In Europe, many of these centers have filed with the health authority what their processes are for collections and which bags they use and things. And if a manufacturer wants a different bag, that may require the hospital to file a change as well. So there are some challenges to organically harmonize. And I think some of these standardization groups can maybe help make this a little bit more formalized. Maybe that is a good transition into sort of our next question. And unless anybody else had gaps discussion, I think we're going to come back to the gaps topic in our third question as well. But maybe we can jump to standardization. Too far. Here we go. So this question is, have you seen improvements, right? We've just started hitting on some of this, and Dawn, you responded to the question related to cryopreservation. Have you seen improvements when it comes to the variability of the requirements and the processes for starting cells for manufacturing?
I've seen a lot of improvement when it comes from the openness and willingness of companies to harmonize and work on standardizing some of these areas. I think, in the last, few years, we've really seen a huge shift from some of these companies to say that it really is worthwhile to look at this. This is making a big improvement to start looking at some ways we can standardize and harmonize. And I think that through the process of developing some of these standards, I think I've seen an improvement of some of these variabilities, shifting towards, being, minimized. I think a lot of the standards that are under development, aren't really fully enacted yet or in place. A lot of them are still either just, just developed or just, published, or are still in the development stages, even if they're towards the end of the development stages. But I have seen a lot of companies starting to try to look ahead in advance of what's coming out and thinking about how they can start implementing some of these things. And I think that that's really moving a huge step forward in minimizing these variabilities. But I think the biggest improvement I've seen is just the willingness and the openness of these companies to work at this and consider this and start being open to implementing these things and having these discussions and being involved in developing these standards.
Yeah, I'll chime in too, because I can reflect. I know in 2019, I gave a talk at the ISCT International Conference in Melbourne, and I talked a little bit about some of the topics of variability and stuff. And I will tell you that in early to mid 2019, this landscape was very different than it is today. There were some efforts starting to talk about harmonization and standardization, but there weren't too many. And the ones that were in place, generally speaking, didn't know about each other. And there wasn't any coordination between them. And some of them were working on the same scope and were going in sometimes two different directions. And we were really getting to the point where it seemed like we might end up actually with more differing standards than there were in fact differing companies, and that we'd be better off if the companies just did it themselves. But I would say in the last 2 years, an enormous amount of effort has gone into sort of aligning and bringing these different groups together. And I actually created a little slide here. This is certainly not everything. It's just a tiny little flavor, and there seems to be buckets of activity that are happening in focusing on whether it's sort of corporate structure, business structure, or topics that are related to the patient or patient follow-up services, topics related to apheresis or starting cells, and then other sort of supply chain topics. And there's a number of different groups out there, and I'm pretty sure that all of these groups now know of each other, and there's a lot of membership of these groups that cross over the multiple groups. There's not a lot of redundancy. Some of the groups are working on similar topics from different perspectives. I know there's the industry working group, the IWG, that Deloitte sort of helps to sponsor. They've got 4 different groups that fit into several of these buckets. One of them is really all about how we approve, certify centers to be able to supply starting cells. Accenture is doing some work also with sort of an industry working group on organizational design and patient services. In Europe, there's several different groups that are working. UK Catapult, which is obviously related to the UK, is a little bit more sort of medical and hospital focused, but has some operations focus as well. And they're working really closely with EBMT that has their Go-Kart initiative. And that initiative is really right now focusing on a lot of the auditing type of question, and actually really driving down into the details of looking at the FACT-JC standard and having companies doing survey of their own expectations to see, well, how closely does this align, and are there things that are additional, either that are in the FACT-JC standard or additional requirements that companies have. The standards coordinating body, SCB, I know I only have 2 of them up here, and that's not giving SCB the credit that they deserve, because if you go to standards coordinating body website, you will see there is an enormous amount of standardization work happening, also partnering with PDA to actually write sort of ANSI standards. Maybe at some point that might transition to become ISO in the future in that type of format. And obviously the cryopreservation standard is one of those. But there's the chain of identity, chain of custody discussions, cell collection standards working group, which is just now kicking off to create an actual standards writing group with the PDA to see how we can write a standard for this. You know, every company is communicating, whether it's through an SOP or a leukapheresis manual or whatever it is, all of our sort of unique requirements for the starting cells. But not only do we have different requirements, we have different ways of communicating those requirements. And so even if there's differences, not only is it hard for the hospitals to track all those differences, they're all in different places in different formats of different SOPs. And so part of this effort might be to try to at least standardize that format. So even if we have different requirements, which may be, right? Each manufacturing process is unique to its own extent. And so there may be unique bespoke requirements, but if a unique requirement is always on section 3.A, it's easier for the hospital to map where those differences are. And so I mentioned that chain of custody, chain of identity, and I think that's also where this BSMA group comes in to talk a little bit about the sort of more supply chain focused as supply chain contributors. And then I think I'm taking the whole panel discussion on this one. Sorry, I'll pass it over to you guys in a minute, but reflecting on the growth that I've seen in the product portfolio that I've been working on for the last 10 years prior to moving to my new job, I very much tried to take the perspective of what I would call controlled flexibility, of really trying to identify what are the critical parameters for our specific product and our specific manufacturing, and focus control there, right? Again, there may be specific things that we need for starting cells for that specific manufacturing process, and how to control that and how to communicate that. But wherever possible, provide the flexibility to the hospitals to use standard procedures, standard processes, obviously with some level of oversight of understanding what those processes are, but not to try to force change where change isn't necessary. And that's kind of been my mantra from the beginning. So maybe I'll turn this question to the panel as well as your thoughts. Maybe I'll go back to the previous to the question slide, back to the panel on where else you've seen improvements.
Yeah, I think when you talk about improvements, you can't, the work of the SCB and those other groups is definitely what pops into my head, and it has been very, very positive. And I expect to see that continuing, and the better coordination between them is certainly helping a lot. One of the things we see is, it really depends where the companies are in their clinical trial phases. When they come into the phase 1 trial, very often it could be the biggest company, it could be small company. They're just, tell us how to do this. How do we do this? Tell us how it goes. As they're more mature, they're getting more specifics, more unique requirements that are problematic in terms of having to train the ACs how to do this and how to go, and how to manage these different requirements. One of the other things we've seen lately is there's been a lot of movement and a lot of influx of people into this industry, as you can imagine, as we are growing. And as those new people come in and they want to provide value, they oftentimes make suggestions and try to push ideas that sort of sometimes move away from standards. Sometimes they move right to standards, and that's very good. That's a very good thing to see. But sometimes they go away. But I think that what the work is doing and putting things out there that people can look at and say, okay, that's how we're going to do it is one of the most important things that I've seen happening in the last 2 years.
I think so. I'm just going to add that I think one of the areas that are issues that really comes up, the clinical centers are dealing with different standards than the pharmaceutical companies are dealing with. So if you're a clinical center, you're dealing with the FACT accreditation standards or ABB or Joint Commission, whatever accrediting body you're using. And yes, so you have the standards that you need to follow. And that really does, it's not so much, well, you are looking at the quality of the products that you are collecting, but it's also the labeling, your SOPs, your change control, your QMS. But then you get, 15 plus different companies that all want a nuance of that. And they're coming at it from a GMP biopharma perspective. And that's where you start seeing, they're tweaking, they want to tweak the labeling, they want to tweak the way the collection happens or the documentation. So I think there has been kind of a meeting in the middle with more and more of the companies. And I find personally that the larger companies are easier to deal with than the small, real small biotech companies that haven't been in the field very often, and especially the ones that actually subcontract out their regulatory or their QA, because then what I'm seeing is those individuals have never dealt with cell therapies or autologous cell therapy products at all. So, I think in one respect, we are seeing improvements in the area. But I still think with there's so many different companies in the market right now, I think for the academic centers, for the collection centers, it still continues to be a challenge. And I think at some point in time, and I think we're getting to that point really quickly, where a lot of clinical sites are really going to say, okay, I can only handle X variability out there. So we're now going to limit who we're dealing with because it's getting unmanageable.
Yeah, thanks. Any other thoughts on this one?
Hey, Peter, it's Gary. I just wanted to add that on the that intersection between the clinical standards and the GMP, because we see that a lot too when we work with smaller organizations, maybe outsourcing some of their subject matter expertise. They coming at it from very much from a GMP biological manufacturing perspective, and they want to see the apheresis collection as just an extension of the manufacturing process and apply the same type of controls and oversight as if it was, a plant, whether it's a contract plant or an owned plant. And I think working with different organizations to understand that this is kind of where the intersection is, and we would, as an industry, be remiss thinking we can force GMP requirements onto centers like the Dana-Farber Center, because that's not the world they're in. But insisting that they do it like they're a contract manufacturer or an own facility is I think somewhere that variability of requirements and processes come, but a lot of it's just more being invented by the industry and people who want to see this as just another biologic, and at least at this step it's not. It's a much different than it is, and that causes some of the friction that's there.
And Peter, I think your approach, looking at the whole process and really applying the changes that need to occur based on risk, that really helps because everybody can get on board with that, especially when you can really explain why you need to do something in a particular way to really limit that risk.
Thanks, Holly.
And I would echo what Alice said there. When we work with clients, that where we start is from a risk-based perspective. Are you doing this because you want to do it, or are you doing this because you understand what the risks are and you're mitigating a risk? And if you talk about that, the partners out there at the apheresis collection are very good at, they know their jobs, they're mitigating risk too. Is it you need to mitigate it your way, or you just need the risk mitigate? And if you identify that up, at least everybody's talking about the same problem.
Yeah.
And I have to say, from a standards perspective, looking at it from that way and developing standards that are really all encompassing and kind of risk-based kind of approach tend to be the best standards because they can be more flexible while also trying to keep things as minimal variability as possible, but allow for flexibility when needed for companies and organizations, so that they can do what they need within their product. And also by doing that risk-based approach and trying to explain why you're doing something slightly different or trying to understand that also helps in the approval process, because that's the exact same kind of questions that are asked when you go for approval. So I think that all around it tends to be the best approach to take when developing standards, but also from a manufacturing perspective as well.
No, thanks, Don. And I see there's also a question which I think is definitely related to this part of the conversation from Brad, where his question is, he's curious how FACT accreditation contributes to the harmonization of apheresis methods. So I thought it would be good to add that here rather than at the Q&A point. I think it's sort of bespoke to directly to what we're in this part of the conversation. I have some thoughts on it, but maybe see if anybody on the panel has thoughts first.
Yeah, I can go. There's definitely very important for FACT accreditation. Of course, we work with the blood centers as well, so AEBB as well, but we think accreditation is extremely important for the industry. We also perform quality audits. And so we have actually done the work of comparing the FACT and requirements against GMP and GTP requirements, the regulations 1271. I mean, and they are, of course, as you would expect, a lot of overlap between those. And that's caused us to look at our audits for accredited institutions and then try to look at ways to reduce the overhead and focus on quality versus just, yes, you have the system, you've proven that by getting your FACT accreditation. So yeah, it's tremendously important that organizations are accredited and continue to follow that, because there's huge overlap with the regulations. And it is an interesting question that I still don't think is 100% sorted out, is when is it GMP, right? I mean, you get different opinions from different people. And if we could just all agree on that, it would make a huge difference in how we approach the apheresis centers and what we do with them, because it's still not, from different companies, we get different opinions.
And I just wanted to add, being a member of the FACT Standards Committee, FACT and JC, we jointly write the standards together. So you not only have institutions in the United States and Canada following the FACT standards for collection, but you also globally around the world, if they are JC or FACT accredited, you are basically following the same standards.
And sort of a note on I mentioned two of those groups that are working, the EBMT Go-Kart, which is doing similar to what Greg mentioned, that Be The Match has already done, is that assessment of companies' own expectations as compared to the FACT JC standard. And then in the IWG, that industry working group, we are looking a lot at the question of auditing and taking the approach that taking input from several of the, many of the companies that are in this field, and sort of trying to see, is there a way that we can leverage as companies, is there a way that we can leverage the knowledge that a site has FACT JC accreditation to either eliminate audits or do a risk-based assessment, to maybe modify the audit scope. There are some companies out there already that are already implementing risk-based assessment and waiving some audits because of FACT accreditation, FACT or JC accreditation. There's others that already are modifying their audit agendas based on what is in the report of a FACT inspection, for instance, and not repeating things that were done. So there are definite efforts individually at some companies to work on simplifying some of the audits, and then through the IWG, and then to some extent through the Go-Kart, to see how can we how can we harmonize? And I think this is where I often use that term harmonize, because when we say standardize, like the standards coordinating body, I think this is a next level sort of rung of really coming up with a standard. And I think sometimes harmonizing might be a little bit easier. And if we can harmonize first, it sometimes will help create the standard. It'll make it much easier to create a standard if most people have already harmonized. So that's a comment on also, I know how FACT is being incorporated into some of these thoughts.
Yeah, I just wanted to mention, so I think Brad's point that he's added of that, you know, FACT is really required and helpful in helping with reducing this variability and some of the harmonization issues, but it's not, you know, completely sufficient. It doesn't solve all the problems. It's not, you know, a perfect fix for everything, but it's really helpful and useful. I think that's an excellent point that he's made. And I think that that's very true.
Yeah, and just to make sure, I mean, FACT has been heavily involved in many of these efforts as well, and been tremendously helpful in that way too. So leading a lot of this and supporting it.
Great. I think maybe that will transition us. I'll move to our next question, which I think flows directly from this. Is we've talked a bit about the efforts that are ongoing, you know, gaps, and then efforts that are ongoing in places where we see improvements and harmonization and standardization starting to occur. So kind of flipping back to the gaps, I'm wondering if anybody is actually seeing any place where perhaps things are, if I might say, backsliding, right? Are there some places where maybe challenges are getting bigger rather than actually seeing sort of progress and improvement? And which of those or other challenges do you think are sort of the biggest sort of continuing or emerging places where we might need some improvement?
Yeah, I think one area that I've seen kind of become a bigger challenge is some of the data management. It seems like there's more and more types of data that are being collected. There's more and more information and formatting that needs to become more and more consistent so that we can have that tech transfer, we can have those common conversations and be able to have those understandings between different organizations and have similar data capture and data formatting and data management. That seems to be actually becoming a bigger challenge as more companies are coming on the scene, more types of data are being collected, and more different systems to collect data are happening. So I think that seems to be one that is increasing currently.
One that we've seen starting a little bit more of, and it's not a standards thing so much as an organizational, is companies are starting to have been splitting maybe the vendor they use to do network onboarding and management, and then with supply chain logistics. And I do not recommend doing that. I would put that, whether, if it's the CRO, have them do both. If it's us, of course, I would love to be us, but don't break those two things apart, because I can tell you from personal experience, the opportunities where they are divided are so much more challenging. The roles and responsibilities are unclear. There's obviously a lot of finger pointing, as you think, and we try to, we have meetings where it's sort of, come to deity of your choice about, hey, we got to stop this, we got to do better, we got to figure this out. And it is an amazing challenge when they split those. So I think you got to look at network onboarding and management almost as the beginning of your supply chain. And so that should then flow right into whoever's managing your logistics and supply chain. That should be the same organization that you can hold completely accountable for those issues. When you break it apart, it is rough.
I couldn't agree with you more on that. We see that sometimes even within one company where different groups are managing different clinical trials for basically the same product. And, team A doesn't know what team B is doing or requesting. So there are different requirements within the same company. And that is obviously very confusing because, from a nurse perspective who's collecting a product for company X, is it to be handled this way or is it to be handled that way? It becomes very, very challenging.
I thought I would again go back to some of the discussion that the points that came up earlier, and I think in some of the earlier discussions or earlier panels. And one thing that came up was talking a little bit about expansion. And it's fairly clear that in the U.S. all of the large BMT centers they're all FACT accredited, right? So they're FACT accredited, which basically means they're all essentially using ISBT 128 labeling.
Mhm.
In Europe, there's a little bit of a more mixed bag when it comes to accreditations, but there's a lot of regulations around stuff. There's the European single code that's used that sort of links to the DIN number, very, very similar kind of thing, even if it's not formally ISBT labeling. I've seen a lot of centers that have their own labels that, it's essentially an ISBT label, but things are maybe just in slightly different places, right?
Mhm.
So high level of standardization. And one of the points that came up in one of the previous panels was expansion to what I would call community centers, right? So think just even in the US, when we move beyond BMT centers, where their BMT reimbursement, frankly, was linked to having accreditation, to community centers that don't have an existing BMT program, but we're looking to the future of cell therapy, CAR T therapy, and potential outpatient procedures, especially as things like cytokine release syndrome and other potential side effects reduce with follow-on next generation products of potentially having more and more outpatient type activities that don't require the same level of infrastructure per se, but yet they don't have the FACT accreditation. They may not have the same level of procedures and systems, and I think that's what at least I've also seen when I look globally, not just at community centers, but different regions and the regulations is, US has a fairly clear GTP, right? 1271 is pretty clear, and it's pretty clear the hospitals need to operate under 1271. They, individual companies may have their own perspectives on GMP or not, but GTP is pretty close to GMP. There's it's a little different, but it's the same flavor of regulation. And in Europe, it's very similar. And in fact, some sites in Europe have to get a GMP authorization. In Germany, they get inspected by the health authority and they have to get a GMP authorization to do the apheresis collection for this, right? But then when I look globally, there are countries where there is not clear regulation on this type of activity, collecting cells, certainly not collecting cells for further manufacturing. And in many of these regions or countries, there's also having a accreditation like Factor JC is not ubiquitous. There may be some, if you're lucky, and in some countries, nobody's accredited. So there's no regulations, there's no accreditations. I see that as a major challenge and a major gap, because when we talk about individual companies coming in and applying their own standard, great. If there's an accreditation and we can get everybody to harmonize and agree that they're going to accept a particular accreditation or accreditation inspection report, maybe we can make the life easier at the hospitals. But when we go into a place where they're not accredited and there's no regulation, you can bet that each company is going to come up with their own list of requirements, and they're going to go to those hospitals and say, these are our requirements. we've got to audit you because you've never been inspected by anybody. And then the next one's going to come, and the next one's going to come, and the next one's going to come. And that is not in the best interest of the patients, because that is going to slow down all of these therapies. It's going to become a bottleneck to progress. And so I think it also kind of gets back to Brad's question, which I think was excellent in the last question, was how can we as companies, essentially as manufacturers, how can we help accreditation bodies such as FACT or JC or AABB or even ISO 9000, right? That's something. Even if a hospital has ISO 9000, at least, you know, there's a step forward with quality management systems. How can we as manufacturers help to facilitate accreditation bodies getting into some of these centers, whether it's community centers or international centers? How can we help get accreditation in there so we're not all individually doing it and creating this wave? Yeah, that's my soapbox. All right, back to the panel.
I want to start with something you said about, you know, some of these products going to independent clinical centers outside of, say, a medical center or a hospital. And that's where, you know, standardization becomes important, but the whole chain of identity, chain of custody becomes even more important, because what are you using for those identifiers to transfer from one entity to a next, and then yet a third or a fourth? So that kind of, you know, makes me very nervous, because then we definitely have to agree on what is that chain of identity to link the initial raw material back to the patient receiving that product. When you know, started going into the healthcare facilities that are totally unregulated and really are kind of ripe for some of these other companies to kind of go in there and try to get their products into these places, because there is no regulations or standards to follow. I think the only thing that the only thing that we can do as a professional society is to really try to make it easier for some of these centers to become accredited, to at least stop the whole process and to really recognize how important that is, so that there is at least a base level of kind of GMP or good standards put in place, whether it's, you know, factor A, B, or ISO. That doesn't, I'm going to say it does matter, but it probably doesn't matter. You have to have good quality systems in place. And I think that's where we need to start to work with some of these countries where they don't really have any guidelines of what are the standards that they need to operate if they're going to have a cell therapy program, especially for these autologous or match related products that need to go back to the same patient.
Thanks, Olive. Yeah.
And Peter, if you don't mind, I wanted to jump in on this, because I think one of the challenges that's maybe a little bit overlooked right now is how these issues and lack around standardization are going to impact downstream processing for the manufacturer themselves. And because the whole conversation of where does GMP start to begin, I think is a great conversation, but I think we can all agree that it's GMP when it gets to the next step in the processing at the manufacturer. And the issue there is without this standardized approach and different ways of doing things, you can end up with real challenges in the cold chain. And just give an example, if you know the packaging that's used is fairly sensitive on how it's used, preconditioning, everything like that, and we've had a client who you almost call them, they create nuisance nonconformances, because if you can't get the pack out right, you're always going to have a temperature excursion on, let's say, a fresh blood collection. So they know it's going to happen. They have to manage through it. And it's setting themselves up, if not for failure, setting themselves up for delay, because they're not managing up farther upstream. So there's challenges that if you don't address it early enough in the process, you're just kind of kicking the can down the road and saying, well, then we got to worry about it a little bit later. And that we see that in something as simple as temperature excursions to something as large as, real questions from the regulators about the quality of the starting material and how was it packed out, how was it transported, have you done significant challenges or, on that to really confirm you have good starting material. And that may not be an issue at the center per se, and the center may not be covered under GMP, but when it gets to your doorstep, it definitely is going to be a problem. And if you haven't set it up in the right, then all of a sudden you're handling all these issues that you really shot yourself in the foot. You've caused problems later down the supply chain. And I think that's a challenge as the industry grows and more volume goes through, that's going to be a larger and larger issue as well.
Yeah. Great. I think I had a slide that had a couple of the things on there that I predicted people were going to bring up, and I had a ... for the conversation. And I think we definitely have hit on a number of those and more. So that's great. I think at this point, unless there's last thoughts on that one, I would move to the audience Q&A. I think we've got a first. Maybe I'll ask the panel if there's any last thoughts on the last question, or if you feel we're ready to jump to this. Okay, we've got agreement. So we do have a first question that came into the chat, which was, would decisions around fresh versus frozen become part of future apheresis standards or harmonization efforts? Thoughts from the panel?
As far as that part of the question, I don't know that there'd ever be standards that would make a decision on that. I think there may be standards that come out as we know a little more about some of the considerations to help you make that decision about which way you want to go and which is better for your process and your needs. But I certainly don't see any standards coming out telling you that you must use cryopreserved or you must use fresh. But I think that it's possible that there could be standards out there, because I think that maybe some of the companies that are coming out and just starting to develop their manufacturing processes may like to have some guidance on what are the considerations I need to be thinking about to make that decision. And how do they affect the variability? Because each of those processes will affect variability in different ways. And how to manage those variabilities based off those processes are very different. And so I think that standards to help you with that area may come out, may be helpful, but I think there'll always be freedom and flexibility, because I think different needs and different products will need different pathways.
Yeah, and I agree with Dawn on that. I don't think there'll be a standard on whether you use fresh or frozen, but I think there definitely needs to be a standard on handling of fresh versus frozen and what you should consider. Because it reminds me of, when, it doesn't seem like that long ago, but when the biologics and the monoclonal antibodies were first coming out, people were putting specific model numbers of packaging and very specific processes into their filing for their cold chain. And then if there was any change, they had a filing issue, right? So to get the industry to start thinking about standard processes, if you're going to use packaging, how should that package be qualified? How do you put it into a process into the filing that gives you some flexibility on changes as standards begin to harmonize instead of painting yourself into a corner? I think we can help out a lot as an organization on how to approach that issues, to give that the flexibility, Peter, you were talking about, but bring some harmonization to it as well. Instead of telling, well, you must use fresh, you must use frozen. If you're going to use fresh, these are the issues you need to consider on the handling. If you're going to be frozen, it's a different set of questions. And these are the risk factors that you have to address in your filing. And this is the best way to go about it, which is very similar what the PDA did on thermal packaging for refrigerated products 20 plus years ago. I think there's definitely a need for that in this in this section of supply chain before it gets so complex, you can never manage the complexity. You can't close Pandora's box at that point.
Mhm.
Yeah, I
Yeah.
agree with Gary on that, and Don too. There should never be a standard that says one or the other. I will say we have a pretty strong feeling that your best results are, for an apheresis product is just have them collect it and send it fresh. You're going to get the best existing material. That's not always practical. So as Brad said, the trend is towards more cryopreservation. But we do have a client who has decided that cryopreservation at the apheresis centers is not working for them. So they're going to centralize their cryopreservation, thinking that will give them better results on the product. So there is, just because it's not just cryopreserve or not, it's where you cryopreserve it and how you do that that can help. So I think there's still a lot to go in this area, but it's definitely been a challenge for a lot of our clients.
I think there's enough to talk about on this topic. It probably could be its own panel discussion
Yes.
Of supply chain and product considerations around fresh versus frozen. Yeah, there's a lot to unpack there, but thanks. I'm cognizant we're almost out of time. I think maybe we have time. If there's one more question from the audience, we could probably try to take that.
I have one question, Peter, for the panel.
Yes, Devendra.
You all talked about variability caused by the clinical and commercial drug developers, and also the variability caused by hospitals. Would the panel provide some insightful direction for the drug suppliers with the objective of reducing the variability?
Well, I think we're all working on that. We're working on the various groups to trying to reach some consensus and trying to harmonize, our needs, both on the pharma side as well as on the clinical side. But I still think, it's going to take a while.
Yeah, I agree with that. I think, as minimize the specialized equipment and then supplies as much as possible, because, once you get into that, then it's a choice of either the manufacturer has to supply it at the time of the, therapy collection or therapy infusion. And that's much more difficult than it sounds. Or you ask the, apheresis center or hospital to inventory something for you, which is almost impossible. They don't have the space, they don't have the capability to do that. So just working with sort of the generic supplies as much as possible is a huge difference in terms of how you're gonna succeed in this business, I think.
Yeah, I had mentioned the concept of controlled flexibility, and I think it's absolutely critical. But it can be easier said than done, right? If you're developing a new product, a new process, you may not know what about the starting cells is the most critical aspect. And so you may not quite know what needs to be controlled. So, it's possible you end up controlling more things in early phase. And then as you learn, in traditional pharma concept, it's critical control attributes, right? You figure out what are your critical control attributes as you go through development. And then by the time you get to commercialization, you're really just only controlling those things. And I think that gets to, in my mind, regulatory strategy as well, is what are you writing in your IND, and what are you going to write in your, say, for the US at least, for your BLA, right? If you write it in your license, and then you figure out that, well, this isn't really critical, we don't really need to control it, but it's in your license, it's now going to be a hassle and a lot of work for you to walk that back. So think really carefully about what you write into your filings around your control strategy, even if there's some things that you're, putting in as controls up front in those early patients, those first batches, until you've really figured out what your critical points are. So that's my last thought on that.
So Peter and the panel, I want to express our eternal gratitude to you for this very historic learning for the entire industry. I really believe this was a milestone for us all to make progress in what appears to be a critical area and a formidable challenge for all of us. So I want to thank you, thank you, thank you.
Thank you.
Devendra, and thanks to the
Thank you.
panel and to the audience and the questions that came in too. So I will stop my share now.